Good evening, everyone. It's already 6:10, and we will start with our session. This is week 8. We are in our week 8, and I'm Amrita, your guide for this interactive session. And we have already completed 7 weeks of live interactive session for the course of animal physiology, right? So we are in our week 8, and today I think this is the last week of discussing the neural system. Next week we'll be discussing another topic of animal physiology, right? So this is the last week of discussion for the neural system, and I today also there are some very interesting questions as we are discussing the questions from the lectures of week 8. I'm hoping that everyone has attended the lectures and have joined the class so that we can discuss the questions in a very active manner, right? So the rule is same. I'll be giving you 10 questions, and you have to answer the answers of those questions in the chat box by seeing the options. So I'll give you 1 minute time, and you have to answer your correct choice in the chat box, and then we'll discuss an correct explanation to that question. It is not a worry if you don't know the answer, try to guess it and type it in the chat box. If it's wrong, it's okay, you will learn something from the explanation. If it's correct, well and good, right? And yes, I want everyone to keep noting down how many correct answers you're giving out of the 10 questions, and we'll know your score at the end of the session. Okay, I guess I'm clear with all my points, and without delaying our session, we'll start with the first question. Okay, so the first question is major population of skeletal muscle is intrafusial fiber, extrafusial fiber, both A and B are equal, or none of the above. I'm repeating the question again. Major population of skeletal muscle is intrafusial fiber, extrafusial fiber, both A and B are equal, or none of the above. Your time starts now. Start answering. Is it not clear for everyone? Is it still not clear? Okay, okay, great. Read the question properly and answer it. Okay? If anyone has any queries, please do not raise the hand but write it down in the chat box. I'll see. Very sure. I want answers from everyone. Not many people are there, and I think I can count how many answers I'm getting, right? So I can see only 4 or 5 people have answered till now. Think, think, take your time, but answer in the chat box. I'm hoping everyone is attending the lectures week-wise regularly, right? Is there anyone who is not attending the lectures? And I'm also hoping that you all are doing your present year assignment questions properly, you're getting full marks. Like, I don't think any of the assignment questions are out of the session. So, and these are very direct questions, of course. And if you attend the lectures properly, I think you can answer all of them correctly, right? Everyone, try to answer the question. Is everyone done? I can see many people are not answering. Why? Why so? I'm not scolding here anyone for giving a wrong answer, isn't it? We all are here to learn the concepts, revise the concepts of week 8, right? practice some questions through the concepts, isn't it? So it will be good if everyone tries to give an answer, even if it's wrong or right. It's okay. People who are joining late, I'm giving them some extra time. We are still at our first question. Try to read the question and answer it. It's a very simple question. We have already discussed about types of muscles in the previous classes. What are the types of muscle? What are the different properties of these muscles? How are they different from each other? Here in this question, we are talking about skeletal muscles. Okay, I can see there is some confusion. Some are saying intrafusal, some are saying extrafusal. What is the correct answer? Okay, it's time up. We'll see what is the correct answer. The correct answer is extrafusal fiber. So whoever has given B as the correct answer is correct. So most of the skeletal muscles are mostly made up of extrafusal muscle fibers. So these muscle fibers are obviously multinucleated in the skeletal muscles, and you can see this is a skeletal muscle, and if you see the core component of it, these are the extrafusal fibers, and also intrafusal fibers are present in skeletal muscles, but the majority proportion is for the extrafusal fibers. So what is the function of these extrafusal fibers? They produce force to move or rigidly fix the body parts. Like obviously skeletal muscles are present in our hands, in our legs, right? And in our limbs basically. So these, and we always use force for doing for any of our movement, for any of our work, and these extrafusal fibers help in doing that forceful work through our legs and hands, right? And if you want to know what is the major difference between these extrafusal and intrafusal fibers, so extrafusal fibers are a bulk of muscle, right? Are a bulk of muscle, and they can generate a large amount of force. That's why they are called extrafusal fibers. And intrafusal fibers are buried inside the muscle, and they contain some receptors, right? They contain some receptors which can sense the signals, and are mainly contain the contractile elements for muscle contraction, right? So I guess everyone is clear with the difference between the extrafusal and the intrafusal fibers. So extrafusal fibers are mostly for movement. They generate force that is required for movement, whereas intrafusal muscle fibers have receptors in it that is required for sensation, that is required for the muscle contraction. They are buried in the muscles. Okay, and majority part of our skeletal muscles is extrafusal muscle fibers. Okay, okay, great. We'll move on to the next question. At neuromuscular junction, muscle is dash and neuron is dash. Postsynaptic, postsynaptic, presynaptic, 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 postsynaptic, or postsynaptic, presynaptic. This is a very easy question, I guess. At neuromuscular junction, muscle is dash and neuron is dash. Your time starts now. Start answering. Okay, some people have given the answer. I can see only two answers. Rest of the people, what are you doing? See, these are very direct questions, and I think, like, if you know it, if you know it, if you don't know it, you don't know it, right? Like, these are very factual questions, not concept based questions. So At neuromuscular junction, muscle is dash and neuron is dash. I can see only 3-4 answers. What are the rest people doing? They don't want to answer or they are still thinking? I want answers from everybody. Guys, please try to be active in the class. Then what's if you're not active, if you're not answering, if you're scared of answering, there is no point in joining the session, isn't it? Don't you think? You're just thinking of knowing the correct answers to each question and then going back, right? If you attend the question, you will remember it for a long time. If you don't attend the question and just know the correct answer, after some days you'll definitely forget because you didn't make an effort to understand that question, isn't it? Is everyone done? Should I discuss what is the correct answer? Only 5 people have given the answer. I can see the population is decreasing week by week, but also whoever is joined, they are also not answering. Is the question too difficult? I don't think so. If it seems difficult for you, you definitely need to go back and attend the lectures, right? Definitely need to go back and attend the lectures so you can be clear with these concepts. Here I am obviously discussing the explanation to all the answers, but if you want to start from scratch and know every bit of it, you need to attend the lectures. We can't discuss all the basic things in this 1 hour session, right? Okay, I'll take it that everyone who wanted to answer has answered. So we'll see what is the correct answer. Yes, whoever has answered, they have answered it correct. It is D option, that is at neuromuscular junction, muscle is postsynaptic and neuron is presynaptic, right? So here you can see this is a diagram of a neuromuscular junction and the neuron is the neuron is the presynaptic membrane and the muscle is the postsynaptic membrane. Right here you can see the muscle fiber is the postsynaptic membrane and the motor nerve has the presynaptic membrane. Presynaptic is the one which is releasing the chemicals and the postsynaptic membrane is the one which is receiving the chemical messengers which has the receptors in it. So if you want to define a neuromuscular junction, it is a junction where a motor neuron and a muscle fiber is present and a chemical synapse is occurring between these two parts, right? It allows the motor neuron transmit a signal to the muscle fiber for muscle contraction. We have already discussed the mechanism of muscle contraction in our previous weeks. So I hope everyone is clear with the explanation of this question. Okay, we'll move on to the third question. RYR and DHPR are dash in muscle: potassium channels, calcium sensors, chlorine ions, or none of the above. RYR and DHPR are dash in muscle: potassium channels, calcium sensors, chlorine ions, or none of the above. Your time starts now. Start answering. Okay, I can see 2 people are answering very fast to each question. So I'm hoping they have learned and come. That's very nice. RYR and DHPR in muscles. Is everyone done? Okay. So if you're saying calcium sensors, is it? Okay. Let's see what is the correct answer. So the correct answer is calcium sensors. Very good who have answered it correctly, who have at least tried to answer it. So here you can see in this figure there are these two sensors called the RYR and DHPR. RYR provides a channel, it has a channel for release of calcium ions from the sarcoplasmic reticulum, whereas DHPR acts as a voltage dependent calcium channel which functions in the skeletal muscle as a voltage sensor, triggering the calcium release for excitation contraction coupling. We have already discussed about muscle contraction, that is this is the diagram of a muscle contraction, and these calcium ions are required for muscle contraction, and these are being released from the sarcoplasmic reticulum by the RYR and are sensed by the DHPR in the skeletal muscle, present in the skeletal muscle, right? So these two are calcium sensors. Okay. We'll move on to the next question. In muscle, calcium is released from. Okay, we already discussed this. I just told now. I think everyone knows the answer. At least this answer, everyone should answer it in the chat box if you are listening to my explanations, right? In muscle, calcium is released from neurons, sarcoplasmic reticulum, cytoplasmic reticulum, or all of the above. I just now told the answer of this question. If you are listening attentively to what I am saying, I think everyone can answer this question, isn't it? In muscle, calcium is released from neurons, sarcoplasmic reticulum, cytoplasmic reticulum, or all of the above. Like today, this week, we have very direct questions, not so complex questions. And I thought everyone will be able to answer it, but still, I am seeing many people are not active in the class. They are they have just joined, but they are not answering the questions. I'm again requesting everyone, please try to be active and answer the questions. So, okay, if it's wrong or right, we are definitely going to discuss the correct explanation. But if you are not active in the class, you are at loss, definitely. Right? This is only an online session. We are not even meeting front to front. And I'm not scolding you people for wrong answers, right? We are all here to learn something from this session. In muscle, calcium is released from neurons, sarcoplasmic reticulum, cytoplasmic reticulum, or all of the. Everyone has said sarcoplasmic reticulum, whoever has tried to answer. Okay, let's see if this is the correct answer. Yes, this is the correct answer. I just discussed it, right? The calcium, our calcium is stored in the sarcoplasmic reticulum and is released and sensed by these calcium sensors during muscle contraction. Calcium is the ion which initiates the muscle contraction in our body. It is a very important ion for muscle contraction, right? We'll move on to the next question. Now, from the nervous system, we have come to, okay, we have, we'll discuss something about the ear anatomy. Which of these is not a bone in ear? Which of these is not a bone in ear? Malleus, stapes, femur, incus. This is a very famous question. I think everyone knows three parts of the ear. I think which of these is not a bone in ear? Malleus, stapes, femur, incus. I guess I can expect 10 out of 10 from some students at the end of the session. Questions are very easy, and if you have attended the lecture, I am sure you can give correct answer to each of the questions we are discussing today, right? Which of these is not a bone in ear? Malleus, stapes, femur, incus. Okay, let's see what is the correct answer. Correct answer is C, femur. Obviously, malleus, stapes, and incus are all parts of the bone, and femur is not a bone in ear. What is femur? Where is it present? Here you can see the anatomy of ear. Here you can see the parts in the options. Incus, malleus, stapes. All of these are bones in the ear, middle ear. These are parts of the middle ear specifically. And femur is a thigh bone. The thigh bone is known as the femur, right? And it is the longest bone present in our body. I guess everyone knows it. Femur bone is the longest bone present in our body according to its length. It is present in thigh, not ear. Okay? I guess it was a very easy question. Everyone is clear with the explanation. We'll move on to the next question. Hair cells in ear have one long hair called. Hair cells in ear have one long hair called stapes, femur, kinocilia, stereocilia. Stapes, femur, kinocilia, stereocilia. Your time starts now. Start answering. These questions will help you to answer the present year assignment questions also. Those questions also must be similar to these questions. So if you are practicing these questions in this assignment session, you will definitely be able to answer the questions, think properly and answer those questions as well, right? You all will be giving an exam at the end of the session. I'm not sure how many people have registered, but yes, if you are attending the lectures and giving the exam, that will be very valuable. Yes, also as I repeat in every class, you will be getting this PPT as well as the recording of this session in my YouTube channel. If any one of you wants to revisit or rewatch the videos, you can definitely go back and watch it in the YouTube channel. Want to know or you want to understand the explanation, you couldn't understand it through this 1 hour session, you can go back and like I'm giving proper notes for each question, so you can always go and watch the session or see from the PPT, right? C. Everyone is saying C. Is everyone done? Hair cells in ear have one long hair called stapes, femur, kinocilia, stereocilia. Okay. Okay, some people are still answering. I'll wait for 1 more minute. If anyone has to answer, answer it. Okay, we'll see what is the correct answer. Correct answer is C, Kinocelia, right? So if you can see the figure here, this is the structure of the ear in which the hair cells are present, and if you can see these hair hair cells, these hair cells are connected to special hair-like structures called kinocelia and stereocilia. So kinocelia, these long hair, out of the three hair, the longest hair are indicated as kinocilium, right? So kinocilium, if you see as one hair, if you say it, the singular form is kinocilium, is a special structure connected to the hair cells. You can see the hair cells of the ear, not of head. It is the hair cells of the inner ears, cochlea, the cochlea, right? It, what is the function of these kinocilium? It acts to aid in depolarization and hyperpolarization of the plasma membrane due to the bending of stereocilia. When these small hairs called the stereocilia bend, they act, they help the kinocilia in polarization of the plasma membrane. Right? This ear also has the plasma membrane. It also undergoes through the action potential and signaling in our ear as well, and these hair cells, the kinocilium and stereocilium in the hair cells connected to the hair cells, take a major part in that action potential in the ear, right? So we'll move on to the next question. Hair cells in ear have multiple short hair called stapes, femur, kinocilia and stereocilia. We have already discussed the correct answer to this question, but still I'll see how many people are giving the correct answer. Please mute your videos. Hair cells in ear have multiple short hair called. We discussed what is the long hair called, and this is question about short hair. And I already told you that, right? Okay, I won't waste much time in this question. We'll see the answer. Everyone has given the correct answer. The correct answer is D. Stereocilia are the short hair connected to the hair cells, whereas kinocilia is the long hair. So, what are in the inner ear? Stereocilia are the mechanosensing organelles of hair cells, which respond to the fluid motion in numerous types of animals for various functions, including hearing and balance. So, these stereocilia function in hearing and balance in our ear. They are about 10 to 15 micrometers in length and share some similar features of microvilli. So their length is around 10 to 15 micrometers. It's very small and share some features of the microvilli. Who can say where is microvilli present in our body? Does anyone know where is microvilli present in our body? Microvilli, microvilli. No? No one knows? Yes, it is present in our small intestine, right? Has some same similar functions means microvilli in our small intestine increases the surface area for absorption. The same function is happening in our ear also. Okay, it increases the surface area for response to fluid motion in our inner ear, right? It increases the surface area for mechanosensation. It increases the surface area for sensation in our ear. for hearing and balance as well, right? We'll move on to the next question. Which of these cells is related to olfaction? Which of these cells is related to olfaction? Hair cells, hippocampal cells, motor neurons, or mitral cells? Your time starts now. Start answering. Which of these cells is related to olfaction? Hair cells, hippocampal cells, motor neurons, or mitral cells? We have discussed about hippocampal cells in our previous sessions, right? Olfaction. What do you mean by olfaction? I guess everyone knows the meaning of the term olfaction, right? Olfaction means smell, is it? I can see only three answers till now. Many people are not active in the class. It's very disappointing. Are you guys joining and going back to sleep? Is it? Is everyone done answering the question? Should I discuss what is the correct answer? Okay, we'll see what is the correct answer. Correct answer is D. Mitral cells. Hair cells, we already saw it is present in the ear. Hippocampal cells and motor neurons are present in the brain, but they are not involved in olfaction. Mitral cells are the ones present in the olfactory bulb of present in our nose and like present in our nervous system that is required for smell, right? There is a olfactory bulb present in our central nervous system that is required for the smell, the things we can smell. It gives information to the brain, and then we can know that okay, this is a good smell, this is a bad smell. So mitral bulbs help in giving the information of to the brain through, and they are present in these olfactory bulbs, right? So you can see in the figure also, these are the mitral cells, this is the olfactory bulb. You can see here, you can see these are the odorants, that is the how can we smell because of the particles present in that smell, right? Those are called the odorants. Okay, we'll move on to the next question. 1 decibel represents an actual increase of dash times in sound energy. 1 decibel represents an actual increase of dash times in sound energy. 0.126, 1.26, 12.6, or 126, 126. Your time starts now. 1.26, is it? 1 decibel is equal to how many times an increase of sound energy? Okay, let's see if it is the correct answer. Everyone has given B and B is the correct answer, right? 1 decibel represents an actual increase of 1.26 times in sound energy. That is, like you can say 1.26 is equal to 1 time. So, if you see, logarithmic scales are used to measure decibels. This is a method of accounting for or measuring an exponentially growing quantity. Okay, so for instance, every 10 dB increase in decibels corresponds to a tenfold increase in sound pressure level. So 10 dB increase in decibels is equal to tenfold increase in sound pressure. Similarly, the question was for 1 dB. So 1 dB is equal to increase in one time of sound energy. One time, that is approximately for 1.26, right? So the correct answer is 1.26. We'll move on to the next question. This is the last question of our session. Cochlea is part of inner ear, middle ear, outer ear, not near, none of the above. Cochlea is a part of. I think this also I have shown in the figure in the ear anatomy when I showed you the ear anatomy. If you have noticed it, I think you can answer it. Cochlea is a part of inner ear, middle ear, outer ear, none of the above. This is the last session. After this, I'll ask you the score. Be ready with your scores. Okay, most of you are saying inner ear. I think someone has said middle ear. One person. Is everyone done answering it? Done? Should I say what is the correct answer? It's a very simple question, direct question. The answer is inner ear. I already showed you this figure. Cochlea is a part of the inner ear, whereas malleus, incus and stapes are part of the middle ear. And the outer ear consists of the auditory canal, pinna and the eardrum. Middle ear consists of only these, whereas in inner ear there is this cochlea, auditory nerve, semicircular canals as well as the eustachian tube. Okay, so the correct answer is inner ear, right? Okay, with this we are at the end of our discussion for 10 questions. And yes, let's know your scores out of 10. Answer it honestly. Obviously, even if you say wrong, I won't be able to guess it. But yes, I expect honesty from everyone. What is your score out of 10? Whoever have been answering for all the questions, they can tell me their scores. It's good to know how people are attending the questions and answering it. 9 out of 10, Arshi. Very good. 9 is a very good score. Anyone else who wants to tell their score? No one? Ma'am, show the 1 and 2 question. 8. 8. Okay. First question was about the skeletal muscle. What is the major population of skeletal muscle? And the answer was extrafusial fibers. Why? You can see the structure here, extrafusial fibers. They are the ones which are the major force generating structure in our skeletal muscles, where and are present in majority in the skeletal muscles, whereas intrafusial fibers are also present, but they are buried inside the muscles, and they have these afferent receptors for stretching, for contraction of our skeletal muscles, right? So extrafusial fibers are involvement in force generation, whereas intrafusial fibers are involved in muscle contraction, and the majority of our skeletal muscles have are the extrafusial fibers. This was your first question, and the second question was, at neuromuscular junction muscle is postsynaptic, whereas neurons are presynaptic. That is, the signal goes from the neurons, from the presynaptic membrane of the neurons, and are received by the postsynaptic membrane of the muscle fibers, right? For causing muscle contraction, for the muscle contraction. Okay, 7 out of 10, 8. No one has scored 10 out of 10. No one. Okay, so Archie is the highest scorer, 9 on 10. Show the seventh question. Sure. Let's see what was the seventh question. Okay. Seventh question was, hair cells in ear have multiple short hair called stereocilia, right? Kinocilia are the long hair connected to the hair cells, whereas multiple short hair are present in are called the stereocilia connected to the hair cells. Here you can see group of 333 cells, and the small hair are called stereocilia, whereas the longest hair is called the kinocilia in the inner ear. These are present in the inner ear, and this stereocilia, the multiple short hairs, are involved in hearing and balance in our body, the fluid motion in our ear, right? Okay, okay. Thank you, everyone. We'll meet again next time in week 9, and I'm hoping and I'm requesting everyone again and again to please be attentive in the class, active in the class, try to answer all the questions, wrong or right, and always come after attending the lectures of that particular week. That will only help you in answering all the questions. Yes, yes, you can definitely ask the question. I'm not sure I can. Yes. I think that yes, when we use a earbud, we like the ear wax, whatever we have in our ear, that goes more like it, like it is. I have read somewhere that ear wax is actually good for our head. It helps in insulating the sensations in our body. And yes, whenever we are using a earbud, it definitely doesn't go till the inner ear, that will damage our hair cells. It is present in our inner ear. We cannot damage our hair cells. You can just forget about it. It's definitely not. It's completely normal to have earwax, and yes, now like after very much research, it is being said that earwax is actually good for our ear, and by using earbuds, we actually push the earwax more inside the ear than taking it out. So No, so it's not advisable to use it actually. Okay. Yeah, yeah, sure. Okay, so I guess anyone has some more questions, or we can end this class here. We'll meet again next week, week 9, and yes, we are completed with our neural system. It was a very long topic to discuss. Next week, we'll have some more topic, and it will be more interesting than the neural system, I guess. You'll see. And yes, keep attending the lectures. And yes, we'll meet again next Tuesday at the same time. Thank you, everyone.